My name is Mohit Kumar from group 317. Today my topic is about cell membrane and the structure of cell membrane. Cell membrane. Cell membrane is made up of phospholipid and its associated protein. Cell membrane is lipid bilayer. We can see this is the structure of cell membrane, which is a lipid bilayer, in which the head part is hydrophilic in nature, main which is water loving, where the tail part is hydrophobic. So hydrophobic, which are mainly lipid soluble. Some cholesterol protein also integrated in the cell membranes. That cholesterol which is intercalated between the phospholipids. And some carbohydrate and protein are also two types: integral protein and the peripheral protein, which attach in the cell membrane. The cell membrane has two leaflet, the outer leaflet and the inner leaflet. The outer leaflet and inner leaflets are slightly different due to their symmetry. For example, the outer leaflets contain the phosphatidylcholine and the sphingomyelin, whereas the inner leaflet contain the phosphatidylethylamine and phosphatidylserine. So both are unsymmetrical and slightly different. Cell membrane also show the property of endocytosis and the exocytosis, because we know the hormones are released by the property of exocytosis, whereas if any bacteria pathogen attack to the cell membrane, then the neutrophil goes and the macrophage comes out to the cells by the process called endocytosis. This show the slightly different asymmetry. This is a whole structure of cell membrane. Cell membrane, for example, this is the structure of cholesterol, and cholesterol some receptor, serpentine receptor are 7 membrane protein structure which attach to the cholesterol. These present in intercalated in cell membranes. So increases the cholesterol content in cell membrane become more packed and the membrane is less fluid. This is a structure how the serpentine receptor look like. For example, if question arise, the proximal nocturnal hemoglobin is due to the defect. The meaning of nocturnal hemoglobin, for example, there is a protein called GPI linked protein, that is called CD55 and CD59. If there is a deficiency of that protein CD55 and CD59, then the patient has bursting of RBC and hemoglobin appearing in urine. That is hematuria, means hemoglobin present in urine. For example, if the question arises, the microneedle is inserted into cell membrane. After the procedure, healing of cell plays take due to the these are hydrophobic interaction. If we insert a microneedle in our cell membrane, then the healing occur due to the hydrophobic interaction in the cell membrane. The cell membrane fluid model was given by the Singer and Nicholson. So its model is called as fluid mosaic model and Singer-Nicholson models. It's mentioned that the protein embedded in the lipid bilayer, there is a specific concentration of lipids, protein and carbohydrate that present in the cell membrane. The cell membrane consists on the surface there is a 42% lipids, 55% protein and the 3% carbohydrate. By weight, we see the ratio of protein 1:1. Squamous cell. Also in squamous cell there is a 76% lipid, and oligodendrocyte cell membrane also has the highest content of lipid in all over the cell. That's why during the nitrogen necrosis the nerve system most affected, because squamous cell and oligodendrocyte contain more lipids. Then question arises, what is nitrogen necrosis? Nitrogen necrosis is a change in consciousness, neuromuscular function and behavior by the breathing compound into the gases. For example, there is a in the patient which has nitrogen necrosis, there is a symptom called confusion and judgments, because the squamous cell and oligodendrocyte cells are affected. On the other hand, highest protein to lipid ratio is found in membrane of inner mitochondria, that is 76% lipid, and parasynaptic membrane due to the receptor of blocking of vesicles. The carbohydrate occupy 3%. Carbohydrate also generate our immune response like antibody antigen interactions. Carbohydrate provide symmetry to the membrane and specify. Lipids like phospholipids, cholesterol, triglyceride, fatty acid, these are the lipids. Cell membranes. Some special protein also present, that is called G protein, which is also called a G protein coupled receptor, which is 7 membrane, that is called serpentine receptor. Also the peripheral protein either side of membrane, they are present on the surface of membrane. A more special class is called GPI linked protein that are attached to the cell membrane via GPI anchor. For example, CD55, that is decay accelerating factor. CD55 is very important because if there is a deficiency of CD55, then there is a attacking of RBC, our immune system attacking RBC, and the bursting of RBC, hemolysis occur, and hemoglobin is released in urine, that is present hematuria. Next, protein and junctional complexes. Desmosomes. Desmosomes, where is desmosome present? Desmosome found in our skin, cervix, at urgent membrane that provide the tensile strength. So desmosome provide the tensile. This is a structure of desmosomes. These are the plasma membrane of urgent cell. These are the anchoring protein present in cell. There is a intermediate filaments and that membrane protein that link the cell. This is the structure of 3D view of desmosomes. Second is gap junction. Gap junction means freely permeable junction between adjacent cell. For example, the molecule move easily without any obstruction that present in gap junction. This is a structure of gap junction. In gap junction, the structure present is called connexon. The connexon is a two adjacent cell. It is a hexameric structure. For example, 1, 2, 3, 4, 5, 6. It contains the 6 structure monomer and it form the hexamer. There's connection present in gap junction. If there is a defective in connection 32, then the disorder is called Charcot-Marie-Tooth disease. It is a demyelitic disorder that affects the gap junction found in squamous cell. For example, if there is a defective in connexon 40 in atrial myocardial cell, then connexin 32 protein form the channel through myelin sheath, allowing efficient transport and communication between a outer myelin layer and interferon of a squamous cell. Third is tight junction. What is tight junction? Tight junction molecule are very hardly move. For it is found in our endothelial cell. Endothelial cell, it is made up of claudin, occludin, and junction. It's mainly claudin and occludin. Claudin form paracellular barrier and pores that determine the tight junction permeability. For example, claudin responsible for permeability. This is the structure of how tight junction work. There is the transfer of molecule very difficult because mucus layer are present on the surface of tight junctions. Occludin, claudin 1, E-carnitine, ZO1, JAM1, catrin, actin. These are the tight junction special protein. Tight junction mainly found in our GIT tract and blood-brain barrier. Blood-brain barrier are that type that separate the blood from brain. This is also found in endothelial cell. Endothelial cells are present in blood vessels. By inflammation, these endothelial cells are damaged. They transport protein in cell membrane. There are mainly 4 type of transport, like pores by passive transport, channels also passive. If there is a carriers called active transports, if there is a pump, are active transports. These are all the structure of cell membrane and their structure. Thank you very much.